name is Fenchen Bao. Today I am going to present a review of the paper Simplifying the Utilization of Machine Learning Techniques for Bioinformatics. Before I start the review, I just want to point out that this is a very well-written paper, probably one of the clearest I have read. I'll definitely learn from the way the authors explain their methods and present their analysis, and hopefully I can emulate that in my own academic writing. So the paper has two very clear goals. It wants to know what feature selection algorithm is the best to reduce dimensionality in bioinformatics data. Now, bioinformatics data, specifically those from DNA microarray, carries a lot of features. For instance, a feature can be a single nucleotide polymorphism, which can amount to thousands in one analysis. So it is apparent that it's a fool's errand to try to include all the features in data analysis. It's because not only will that slow down the analysis, but will also introduce a lot of noise or collinearity that could jeopardize the analysis accuracy. Hence, finding a good feature selection algorithm is very important to handle high-dimensional bioinformatics data. Now, the paper's second goal is to answer the question, what is the best classifier to deal with bioinformatics data? We are talking about using a classifier to make judgment calls based on the data. Now, typical example is given the data of, say, 100 genes in a person's skin tissue, we want to know whether the tissue is cancerous or not. So a good classifier can increase the accuracy of such judgment calls and is thus also paramount to bioinformatics research. To conduct experiment, the paper first obtained the datasets, which consist of 25 published bioinformatic datasets generated by DNA microarray or mass spectrometry. All the datasets are binary labeled, which means each observation in the data belongs to one of two classes. These two classes can be cancerous or not cancerous cells, or one type of cancer versus another type of cancer. To analyze the performance of feature selection algorithms, the paper selected 24 filter-based feature rankers. Typically, there are two types of feature selection algorithms. One is filter-based, which can be conducted independently from the classification, and it also runs fairly quickly. Now, the other one is called wrapper-based, which requires actually using a classifier to help with feature selection. The wrapper-based feature selection is slow in computation and very limiting in its generalizability. Therefore, the paper went with their filter-based route. Feature ranker means that the feature selection algorithm produces a ranking of all the features, and the researcher can decide however many highest ranking features to include in the analysis. So among the 24 filter-based rankers, 7 are commonly used, including chi-squared, information gain, gain ratio, relief F with weight by distance, symmetric uncertainty, SCVM non-recursive attribute evaluation. 11 of them are threshold-based, which are also developed by the same team writing this paper. They include mutual information, F-measure, odds ratio, deviance, geometric mean, Kolmogorov-Smirnov statistic, power, probability ratio, Gini index, area under the receiver operating characteristics curve, and area under the precision recall curve. And finally, the rest 6 are first-order statistics based, including signal-to-noise, Fisher score, fold change difference, Wilcoxon rank sum, Welsh t-statistics, and significance analysis of microarrays. The paper chose 4 different numbers of features for testing. This means each feature ranker will be used to generate 50, 75, 100, or 200 top-ranking features for analysis. There are good reasons for setting the bounds of the feature size. The lower bound is set to 50 because previous research has shown that this is the lowest number of features necessary to construct a meaningful classification model. The higher bound is set to 200 because previous research has found that people just cannot interpret the result of classification if there are more than 200 features to reason with. Regarding the classifiers, the paper tested 6 classifiers. They are 5 nearest neighbors, multi-layer perceptron, naive Bayes, support vector machine, logistic regression, and random forest with 100 trees. The experiment procedure went like this. Say for feature ranker 1, we generate the top 50 features for each of the 25 dataset. We then pass the transform the data to classifier 1, which goes through 4 rounds of 5-fold cross-validation to compute the performance of the model. Now the metric for performance is area under the receiver operating characteristic curve, or AOC. AOC describes how the model balances false positive and true negative rates at different decision level. A higher AOC value indicates better model performance. Now the AOC of 4 rounds of cross-validation is averaged as the output of the performance of classifier 1 relative to dataset 1. And then we obtain this average AUC for all of the 25 datasets, and then we average them again to obtain an overall mean AUC value. And this mean AUC value, the overall mean AUC value, represent the model performance given feature ranker being feature ranker 1, 50 features, and modeled by classifier 1. So the paper repeats this entire procedure for all the combinations of feature rankers, feature size, and classifier, which amounts to 24, because 24 rankers times 4 features, times 6 classifiers, or 576 data points. And all these data points are listed here in these 2 tables. The bold values, so we have some bold values. The bold values are the best performance of a classifier under a feature. So for example, this is the best performance of classifier SVM under feature size 50 across all the feature rankers. The italic values, which are actually quite difficult to see, but for here, let's say this actually the italic value. So this is the worst performance given a classifier, a feature size, and across all the rankers. The last row is called range, which represents the difference between the best and the worst performance for each classifier and under each feature size. From this data, we can observe a couple of things. We can see that the feature ranker SVM ATT has more incidence of best performance than any other rankers. However, whether this is statistically significant remains to be analyzed. In addition, judged by the magnitude of the best performance, we see that random forest 100 has the best performance among all the classifiers. But again, whether this is statistically significant remains to be seen. Finally, look at the range row. We can see that not only RF100 has the best performance, but its range is also the smallest among all the classifiers. Now, a small range, or we say the small difference between the best and the worst performance, means that RF100, this classifier, is very resistant to the change in feature rankers. So now to examine whether RF100 is really significantly the best performance, then we need to conduct a one-factor ANOVA. So the paper does a one-factor ANOVA to examine whether the performance of all classifiers are the same. And the ANOVA is carried out with the classifier being the single factor and with the feature size fixed. And the results show, circled here, show that the probability of all classifiers having the same performance is much lower than the conventional threshold of 0.05. Hence, the paper concludes that the performance of all classifiers is not the same. Note that the feature ranker is not considered in this analysis. Next, to elucidate how classifiers' performance differ from one another, the paper conducts two-case honestly significant difference, or HSD test. And the results show that the average performance and the confidence interval for each classifier under each feature size. So you can see the circles are the average performance and we have the bar indicating the confidence interval. And it is clear that RF100, this one, this one, this one, and this one has the best and significantly the best performance compared to the others. Similarly, we can also observe that ALR, or logistic regression, has significantly the worst performance among all the classifiers. And the performance of MLP here, MLP, 5, NN, and SVM, they are in most cases quite similar to each other as their confidence interval overlap, except for this case, under feature size 200, we see MLP has significantly better performance than 5NN. Moving on, the paper examines whether the performance of all feature rankers are the same. So similar to classifiers, this is a one-factor ANOVA and is conducted with the single factor being the feature ranker. Here we have both the feature size and the classifier fixed. So the results shown here is specific to RF100, which is also the best classifier. We can see that the probability, circled here, of all feature rankers having the same performance is higher, much higher than the conventional threshold of 0.05. Therefore, the paper concludes that the performance of different feature rankers are not significantly different. In other words, the seemingly top performance from SVM ATT is actually not significantly different from any of the other feature ranker. The paper further pointed out that this result is repeated for all the other classifiers. Therefore, overall, we can say that none of the feature rankers had significantly different performance than others across all feature size and across all classifiers. In other words, the choice of feature ranker does not matter. In conclusion, the paper answers two questions it raised at the beginning of the paper. So what feature selection algorithm is the best? And the answer is all feature rankers that's tested have similar performance, and no one is significantly better than the others. Question 2, what classifier is the best? And the answer is random forest with 100 trees, and this has been verified by statistical testing. And that's it for my presentation of the review of this paper. Thank you very much for watching. Have a nice day.